Okay, so today we're going to start talking about cell division. And today we're going to talk about DNA replication, which you'll see some similarities from transcription to DNA replication. It's a little bit different. And then on Monday, we're really going to spend time talking about mitosis, which is the actual process of cell division. So this is Chapter 8. It's pretty short. It's really just these two concepts, DNA replication, which is today, and mitosis, which is going to be on Monday. So cells divide and cells die. That's really what this chapter is about. So organisms must reproduce for a species to exist. Some species will reproduce asexually, and that is a process of reproducing by replicating genetic material and splitting. So some organisms don't have sex. Bacteria, some protists, some eukaryotes are like this. So there are protists that will just divide, they'll split in two, and they'll have two offspring. There are hydra that will actually start growing another version of themselves off the side, and they'll split like this. Plants do this. So bromeliads down here, they'll grow, they call them pups. And after the bromeliad flowers, you'll just see these little bromeliads growing off the base of it. You can cut them off and stick them in the soil, and they'll grow just like that. So we have lots of plants that do this. That's asexual reproduction. So it's a very actually convenient, very easy way to reproduce. It's a very successful way to reproduce because you don't need a partner. You can pass on all of your genetic information to your offspring. So if you are pretty well adapted to your environment, they will probably be pretty well adapted to their environment. It doesn't require a high concentration of organisms, and it's very rapid. You can just split off a bit of you. There are organisms that if you cut off part of them, that'll grow into a new organism. So sea stars, so starfish, they're like this. If you cut them in half, you'll have two. There's some organisms like this. So that's asexual reproduction. So the opposite of asexual reproduction would be sexual reproduction. Sexual reproduction produces offspring by combining the genetic information, the genetic material from two parents, and this is what we'll be focused on through the next chapter, after the next test. We'll talk about exactly how things like sperm and egg, gametes like sperm and egg are produced. So, and after sexual reproduction, you end up with one single cell called a zygote, and the zygote has to then divide to make more cells. So the way humans reproduce, we have males and females. Males produce sperm, females produce egg, and we'll talk about this process of meiosis in the next chapter, in chapter 9. Sperm meets egg, you get fertilization. After fertilization, you get a zygote here. And that zygote is a single cell. So somehow you, at some point, went from being a single cell to being a multi-cell organism, like you are today with thousands and thousands of cells in you. So that process of the egg dividing and growing into an adult is called mitosis. So mitosis is division. Reproductive cells, sperm and egg, are produced through a process called meiosis, and we'll talk about meiosis in the next chapter, which is going from a multicellular human to making a single cell, which actually has half the number of chromosomes, and we'll talk about that in more detail later. So this chapter is about this process, going from one cell to many cells, and also something that you do right now, which is you replace cells that are lost. Okay, so mitosis allows a fetus to grow into an adult, and meiosis is going to be the process that makes sperm and egg cells. Okay, so mitosis actually has many different roles that it plays. So it allows, first off, an organism to grow and develop. So it allows you to go from a single cell to a fetus, from a fetus to a child to an adult. It allows you to grow, add numbers to the number of cells, develop new parts, so grow your brain cells, grow your muscle cells. So they're all made through the process of mitosis. It also allows you to repair tissues. So when you get injured, when you cut yourself, the way that your body repairs that injury is through mitosis. It allows some organisms, we can't do this, to regenerate lost body parts. So if you cut off an arm of some reptiles and amphibians, they might grow back. This is how we have snow crab. So you're able to pull off one of the claws of a snow crab and that claw will grow back. So that process of regenerating lost body parts is also mitosis. And it actually allows some organisms to reproduce. So asexual reproduction is done by mitosis as well. So those bromeliads that produce the pups, they are producing those cells through mitosis. Those cells then can become independent living organisms. So mitosis has lots and lots of roles. So that's the cells divide part of this chapter. There's a little section that I'll talk about next about cell death, but we're not really going to focus on it that much. Cell death, or programmed cell death, is called apoptosis. This is not accidental cell death, like you cut yourself and you destroy some cells. Cells can die in predictable, programmed ways, and this process is called apoptosis. It's cell death. That's a normal part of development. So, for instance, the feet of a duck versus the feet of a chicken. A duck's feet start to grow, and there are cells that connect the toes, and those don't disappear, and then you end up with webbed feet. For a chicken, those cells that are in between the toes actually will die back, and that allows them to have individual toes. And it's the same thing with us. As your hands are growing, the cells that would be between your fingers have died off during early development. If they don't, then you might end up with connected digits or webbing, and that does happen occasionally when that programmed cell death does not occur. So there are some people that will have webbed toes, or at least one webbed toe or one webbed digit of some sort. So programmed cell death is a normal part of development, and it is a way that we've been able to develop things in the way that we have. That's called apoptosis. Okay. So cells need to divide in predictable ways. Cells also need to die in predictable ways for an organism to grow from that single cell into a multi-cell organism. So when a cell divides, it needs to copy all the DNA that is present inside that cell. So we haven't gotten into genetics yet, but we have talked about what DNA is used for. DNA codes for proteins, and you probably know by now from the last chapter and from some of the things we talked about that you have the same DNA in virtually every cell in your body. There's a few exceptions. There's some white blood cells that have slightly different DNA. Your red blood cells are missing a nucleus. Your reproductive cells have half the amount of DNA, but every other cell in your body has the same set of instructions as the other cells in your body. So in order to make new cells, you need to copy your genome, so all of your genetic material. So before any kind of cell division, mitosis, meiosis, or binary fission, which we'll talk about what that is as well, all of the genetic material must be duplicated. Genomes are often organized into chromosomes. This is, again, something we haven't quite talked about today, or talked about yet, but we'll talk about today. Chromosomes is simply a portion of your DNA. All your DNA in your cells is split up into packets or volumes, and a chromosome is one volume of DNA. And we'll really talk about this a little bit more in depth in the next chapter and in the genetics chapter, but you'll see this word chromosome a bunch today, and that's just one little packet of DNA inside your cells. Okay, so the first thing we're going to learn about before we talk about actual cell division is how does a cell go about duplicating its genome? How does it go about copying its DNA so that it has 2 copies, so that when the cell divides, it can send 1 copy to each of the daughter cells, each of the new cells. So just to get you thinking about how this is, we talked about DNA in the last chapter. Here is a short little strand of DNA. What would be the complementary strand, so the opposite strand that would be bound to this? Hopefully this is a very easy question for you. Since the first question was so hard, this one should be pretty straightforward. All right, last few. All right, almost all of you chose B. So almost all of you got it. Oops, that's not working. Oh, it's over here. Okay, yeah. So B, TGATC is complementary to ACTAG. So that was relatively easy for you, hopefully. You just used the base pairing rules that, the T pairs with A and G pairs with C. Right? So if you didn't get that, then you need to remember this, pairing rules. So very straightforward. So this might give you an idea of how a cell might make new copies of DNA. So if you had a chromosome that had ACTAG on one side, it would have TGATC on the other side. If you were to split the chromosome into 2, so that one half was on one side and it was separated from the other half, you could use each of these strands as a template. So if you had this side, you would add a T, a G, an A T, and a C to this. And if you had just this side, you could add a C, a T, an A, and a G, and you'd end up with 2 new double-stranded pieces, right? So you would have just split your double strand of DNA, and you've used each strand as a template. This is called semi-conservative replication, and this is how chromosomes are replicated. Chromosomes will use themselves as a template. They will split in half, and then each side will be used as a template to make a new strand of DNA. So in the new DNA, what semi-conservative means is that half of it is old, the dark blue, and half of it is new, the light blue. So should make sense, and this is how it happens. So we're going to talk about the actual mechanism for making this work as we go through. So it's called semi-conservative replication. So in order for this to happen, what's going to go about is we need to start with our double-stranded DNA. So all of our DNA that's in all of our chromosomes in our cells, we'll go through this. The first step will be separating the 2 strands. So we need to break the hydrogen bonds between the double strands to break them into 2 separate strands. And then we need to use each strand as a template using the base pairing rules. Wherever we see a T, we put an A. Wherever we see an A, we put a T. G, we put a C. C, we put a G. To make a new strand on each side, so the light blue here. And then we'll end up with double-stranded DNA molecules consisting of 1 parental and 1 daughter strand as a result. So that's basically the process we're going to go into. What I'm going to be talking about through the next slides are the names of the different enzymes that make this happen. So we're going to talk about the enzyme that separates the strands, the enzyme that adds the different bases. And there's actually several steps to adding the bases because it can't just randomly start adding bases anywhere. So we'll talk about all the different enzymes and the steps that this happens. We'll also talk about why what happens on one strand is different from the way it happens on the other strand and why that is as well as we get into this. Okay. So here are the enzymes that you will be familiar with by the end of today. So there's an array of enzymes that are required for this process. First one is called helicase. Helicase will actually unwind and separate the two strands in the double helix. So helicase acts like a zipper. It's going to unzip the DNA double strand. There will be binding proteins that stabilize the strands, but we're not going to really talk about them in any detail, but they will be pictured on the next few pages. Next few slides are binding proteins. Because when a strand is single-stranded, it's actually very unstable, so it needs to have some kind of protection to bind it. We're going to talk about an enzyme called primase. So we'll look at this in a little more detail on the next few slides, but DNA cannot be manufactured just anywhere. It needs a starting point. And the starting point for DNA synthesis is actually going to be a little bit of RNA. So primase is going to add something called an RNA primer, which gives our DNA enzyme a place to start. So if you remember back to RNA synthesis, we were able to just make RNA from a DNA template without a starting point. So RNA is different in DNA in that sense. DNA needs a starting point. So the RNA primer is going to be the starting point. So once we have a primer set down, an enzyme called DNA polymerase will add new DNA nucleotides. So following the base pairing rules, DNA polymerase will add new nucleotides. And then finally, there's an enzyme called ligase, and there will be, on one of the strands that we build, a gap that needs to be filled in. So a final little space that needs to be glued together. And what ligase does is it creates covalent bonds between adjacent DNA segments so it can stitch together two fragments of DNA and glue them together. Okay, so helicase, primase, DNA polymerase, and ligase are the main enzymes we're going to look at. And there are also things called binding proteins that simply stabilize the DNA as this happens. All right. So, where this is going to happen, your DNA is a double strand. And there are many of these found along a chromosome, but there are these locations called origins of replication, and it's a spot in the DNA where it's a little bit easier to open up the DNA. So this would be called the origin of replication. And what's going to happen at the origin of replication is that the DNA is going to form a little bubble and start to unwind. And what actually first originally starts this bubble is something we're not going to talk about, but we'll talk about what happens next. So most of these pictures that you're going to be looking at on the next few slides are actually just looking at this as if it was just half of it. So picture that. I'll come back to this kind of overall picture of DNA replication at some point, but we're going to be focused in on just one half of this bubble that's forming in the DNA, and actually what's going on right here where the DNA double strands are unraveling. So just to give you a little bit of perspective here. So once this origin of replication has opened up, helicase, that enzyme that unwinds and unzips the DNA, will bind to the DNA and start moving along the double-stranded DNA, unzipping it. Right. And this region here, where helicase is opening up the double-stranded bits, is called the replication fork. So you'll probably see that word. I use that word sometimes. So replication fork are those words. So replication fork is basically where the double strand is opening up. That's this location where helicase is unwinding. Okay. So helicase has now unwound and unzipped a region of DNA. The next thing that happens is that primase is going to add a short sequence of RNA. So that's this green bit right here. So DNA polymerase, the enzyme that's going to be making DNA, cannot start making DNA anywhere. It needs a 3' OH. So on the 3' end of a nucleotide, it needs an OH to perform that dehydration reaction to start adding DNA. RNA does not have that restriction. RNA can be made on any DNA template, pretty much. So primase is able to add RNA that's complementary to the DNA strand to give DNA polymerase a place to start. So you get this little RNA primer here. Okay, so remember how, if you remember back to DNA, how DNA and RNA have to be made. Do you remember what direction DNA and RNA are made? Yeah, 5' towards the 3' end. So here you've got the 5' end of your primer. Here you've got the 3' end. DNA is going to be made in that direction on this strand. If you look down at this strand, though, which direction is DNA going to be made? Towards the replication fork or away from the replication fork? All right, it's going to have to be made in this direction. This is one of the quirks of DNA replication, is that on one strand, you can be making it in the same direction that helicase is moving, but on the other strand, you have to make it in the opposite direction as which helicase is moving. And we'll talk about the consequences of that as we go forward here. It's always being added to the 3' end. So here's the 3' end. Here's the 3' end. You're going to add on to the 3' end of the growing strand.
So nothing can be added to the 5' end?
No. Not so far as I've told you. So there's one exception that comes at the very, very end of this whole process. But yes, in terms of RNA polymerase, DNA polymerase, primase, they always manufacture things by adding on to the 3' end. We will talk about an exception enzyme at the very end. Yep. Okay, so this strand is being made that direction, this strand is being made that direction. Okay, so now primase has added our 3', or added our primer, giving us a 3' OH. The next thing that'll happen is DNA polymerase is going to bind and start making DNA. Okay. So DNA polymerase has started at the end of that primer and made DNA in this direction on this strand and in that direction on that strand. DNA polymerase will add new DNA nucleotides in a 5' to 3' direction that are complementary to the template strand. So this enzyme here starts at the end of the primer and starts adding on to the 3' end. So the next nucleotide would be added on here, and then here, and then here, as it goes that direction on that strand, and here it's going in the same direction as helicase. Okay so far? So we had helicase unwinding, primase adding a primer, and now DNA polymerase adding DNA that is complementary to the template strand as it moves along. Okay, so now here's where things get a little complicated because we have two things happening in different directions on the two different strands. So what's going to happen to the replication fork? Which direction is it moving? Moving that way, right? So this is going to continue to open up in this direction. And as it continues to open up, what's going to be happening up here? Yeah, it just continues making DNA. It can just keep going in that same direction. But what's happening here? All right, it's making DNA in this direction, so there's going to be a gap here, right? Okay, so it's like my analogy here, which some of you might get and some of you might not, is let's say you have fake fingernails. Your fingernails are opening or growing. There's that gap that forms at the back end of the fingernails, right? That's the same thing here. It's growing away, and there's going to be this opening up. So what you have to do is fill that in, and we're going to talk about how that happens in a bit. So we have different names for these different strands because they are doing different things. The top strand is called the leading strand, and the leading strand is created in the same direction as movement of the replication fork. So this is this top strand here. And it's pretty straightforward. It's going to continue making DNA as helicase keeps moving down the double strand. So as it unzips, this just keeps following it. So it's following the helicase as it goes. So that's called the leading strand. The opposite strand, the more complicated strand, is this one on the bottom, and it's called the lagging strand. So the leading strand is made in the same direction. The lagging strand is created in the opposite direction and thus must be made in a series of fragments. So as helicase moves this way, it's going to open up a gap in the backside of it. So let's say this was the gap here. Let me move up a board so I have a little bit more to see here. Okay, so here's your lagging strand. Here was the first fragment that was made. Here was the first primer that was put down, and it made DNA in that direction. Here was a gap that had opened up behind that primer as helicase moved. So all that's going to happen is that it puts down another primer, and it makes DNA in that direction. So what these little fragments are, each with a primer and a little bit of DNA, are called Okazaki fragments. I'm not going to require you to remember that name, but they were named after a researcher that discovered how that worked. So he was named Okazaki. So this is an Okazaki fragment. So what we've done is, we've made now two short little bits of DNA that now have two little short bits of RNA on them. And this is not something we want to leave like this. So your cells don't want to leave it like this because otherwise, gradually over many, many years of cell division, most of your DNA would end up being RNA if you left it like this. So you need to take this RNA and remove it and replace it with DNA somehow. So that's what makes the lagging strand a little bit more complicated than the leading strand. Leading strand just has DNA, DNA, DNA all the way through. The lagging strand has these little fragments of RNA that need to be replaced. So what happens as soon as this polymerase gets to the backside of this little fragment of RNA, it's going to replace the RNA with DNA. So in reality, it's another polymerase, but we're just going to say DNA polymerase then replaces it. There's actually a separate kind of polymerase that comes on and pulls out these RNAs and replaces them with DNA. But for the purposes of our course, just say DNA polymerase will replace the RNA with DNA. So it's going to remove these fragments bit by bit. Okay, so far? So we've now filled in this RNA bit with DNA. Problem is that you can't add anything to the 5' end, and you've got a 5' end of DNA right here, and you've got a 3' end of DNA here, and there's going to be a gap between them. And this is where that final enzyme comes into play. When after the RNA is replaced, ligase, so that final enzyme, is going to create a covalent bond sealing that last gap up. So this kind of space here that's between the DNA and the DNA will be ligated together, and you'll end up with a continuous strand of DNA there. Okay, so that's the process. So helicase unwinds the DNA. Primase puts down an RNA primer. DNA polymerase fills in DNA. On the leading strand, DNA synthesis is going in the same direction as helicase, so it's just going to continue as helicase moves. On the lagging strand, as a gap opens up behind the primer, a new primer will be put down. A short strand of DNA will fill in that gap, and then the RNA primer from the previous fragment will be replaced with DNA, and then ligase will glue together the final ends. So ligase seals the phosphate sugar backbone. Okay, so that's the process of DNA replication, and this is going to copy all of your chromosomes this way. Okay. Okay. Questions so far? You guys okay with this? All right, I'm going to show you a video of it in a minute or two. But before we get to that, I just want to go back to what I was originally talking about, this origin of replication. So this is not happening in just one spot. It's not, otherwise it would take forever for this to happen. You have lots and lots of base pairs. Your chromosomes are huge. This would take a very long time if it only occurred in one location or if it started at one end of your DNA. So this is happening simultaneously at multiple locations called origins of replication. And those origins of replication are locations that will open up. So here are 3 origins of replication, and it's going to open up a little space in all of them. And in all of them, you're going to get synthesis in multiple directions. So let's say, just because I can reach this, say this is one big origin of replication. What's going to happen here is polymerase is going to, let's use green here, lay down a primer here, lay down a primer here, and initially DNA is going to be made in this direction and in this direction. So those are what are called the leading strands. There's a leading strand going towards this helicase, which is headed in that direction, and there's a leading strand going towards this helicase going in that direction. This is where this gets conceptually a little complicated. As the bubble opens up a little bit more, there ends up being a gap here on both sides. So a primer will be added here, a primer will be added here, and DNA will be made in this direction. So this would be the leading, and this would be lagging. And on the top here, this would be leading, and this would be lagging. So this is why we only looked at half of it, because it's a little bit easier to comprehend when you're only looking at one half of this, because you have the leading headed towards helicase, the lagging headed away. But there's also the other half where you have the leading heading towards helicase and the lagging heading away. So the ultimate take home from this is that if the DNA is being made in the same direction as helicase, it's the leading strand. If it's being made in the opposite direction as helicase, it's the lagging strand. So on the left side of this bubble, the leading strand's on top and the lagging's on bottom. On the right side of this bubble, the leading strand's on the bottom and the lagging's on top.
So are both helicases opening up the opposite direction?
They're going in both directions, yeah. So at each of these bubbles, so if you look up here, you've got a helicase here going in this direction and one here going this direction. And you've got helicase like this. The helicase, once it's opened up the whole strand, it falls off. It's done. And then your leading strand, so if there would be a lagging strand here. So there would have been a lagging strand being made here. And ultimately, it's a really good question, because ultimately this leading strand is going to reach the back end of that lagging strand, and it will replace this RNA with DNA. That's how this original primer is eventually replaced. So this lagging strand reaches the back of that, and eventually you fill in all of the gaps. Right?
And then ligase.
Ligase, yep. So ligase will function right here when this leading strand reaches the back end of that lagging strand, once the helicases come together, then it'll replace the RNA and then ligase will glue it together. So it all fits together in the end. We really only have this terminology if you're looking at just half of the bubble focused on one helicase. So when you're talking about leading and lagging, you're really focused in on that helicase. But there's a bigger scale thing going on here. That's really what I wanted to get at from this. All right? Okay. So here is just another view of this with the fragments put in. DNA replication is occurring in two directions. And you'll notice you have Okazaki fragments here, Okazaki fragments here. And that's basically what I just said, right? Okay. So last thing I want to do, and I'm going to show you a quick little video after I show you this, is to show you reality. So we've been looking at this in this kind of exploded, very easy to comprehend version of this, and that's how it's the easiest way to learn about it. But in reality, these are not completely separate enzymes. They're actually all tied together into something called a DNA replication complex. Helicase is actually connected with these little connecting proteins to the DNA polymerase. So it's like helicase has two little arms. One is going to be running the leading strand, and one is going to be doing the lagging strand. And it runs along as this protein complex. So the proteins are actually attached in some way, which is crazy. This is something I actually didn't know this until I started going over this again, because I don't think we even ever covered this when I was in undergrad. So the video I'm going to show you actually shows you how this works if we were to model and have an animation of what's actually going on with the protein complex. So you're going to see helicase attached to two polymerases, and you're going to see the leading strand happening continuously and the lagging strand happening in these little loops. On the video that I'm going to show you, it's actually inverted, so the leading strand will be on the bottom and the lagging will be on the top. So let me see if I can get this going. There's actually some audio, which...
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
All right, so that is the simplified version of this. Yeah. So this is where we're going to stop because we start getting into mitosis in the next few slides, and we'll start that on Monday. So that's the process of DNA replication, and that's going to occur before cell division.